And we're back. And as we go over these problems, we need to have some uh, familiarity with our terms. And so I wanted to set the stage for our genetic problems. Now, some of this should be somewhat familiar. My guess is that you have done this at some level before, maybe in your middle school days. So let's go over some terms. First of all, different expressions of the same gene is called an allele. So, for instance, eye color, you could have blue eyes or brown eyes, hazel eyes, green eyes. Those are the majority of the eye colors that we see, some level of them. So eye color has different alleles. They're still eye color, but they have different versions. Blood type is the gene, but they have different expressions. Type A or type B or type O or type AB. So we have different alleles that then cause different genetic conditions. Now, each person has two copies. Remember, we get one from mom, one from dad. So if you have two of the same allele, we call that homozygous. If we have two different alleles, that would be heterozygous. And so you can see this diagram is straight from our textbook. If this is a pea plant and they have one wrinkled allele and another wrinkled allele on the other chromosome, that would be homozygous. If one chromosome has the wrinkled allele and the other chromosome has the round allele for the pea the pea seed, then that would be heterozygous. Now, some alleles get expressed preferentially over other alleles, or they get expressed whereas the other one is not. We call those dominant alleles. In a dominant allele, you only need one allele because that gene gets expressed. That whatever that protein is, we make it. So if you have type A blood, you're going to make that type A protein. We'll talk about that next week. Now, dominant alleles are usually expressed with a capital letter. We understand that shows that it is a dominant allele. Recessive alleles, you need two of them in order to see that. Otherwise, if you have a dominant and a recessive, the dominant trait is seen. Now, one quick word, dominant does not mean it's stronger. Dominant does not mean it somehow overtakes the other one. That simply means that there is some signal or some way that it gets expressed that we see it, whereas the recessive, we only see it if the dominant isn't there. So the dominant, we use a lowercase letter to represent that. The other two terms that we're going to look at would be genotype and phenotype. Genotype is going to describe the arrangement of those alleles. Homozygous and heterozygous would be the genotype. Do we have big A, big A? Do we have little a, or do we have big, little? That's the genotype. How would we describe that? Phenotype would be what that individual is or what it looks like, the physical expression. Do we have a black Labrador retriever? Do we have a chocolate Labrador retriever? Do we have type A blood? Do we have blue eyes? Those are all phenotypes. So, for instance, in my example, big Y, little y might be the genotype for the yellow pea pods, but its phenotype would be that it's yellow. So the phenotype is what we see or can examine. Genotype is the arrangement, whether or not we have big, little, big, big, or to be more proper, homozygous or heterozygous. So I'm going to have you guys pause this. Let's practice some of our vocabulary with this slide, with this problem. Go ahead and hit pause. And as you guys went over that in class, hopefully you reviewed some of your terminology and you feel a little bit more comfortable. Practice makes perfect. We're going to keep giving you some practice problems throughout this. So this is a true example. These are the genes. This is how Labrador Retriever coat color works, at least for the black and the chocolate versions. A black lab versus a chocolate lab. Yellow labs have a like another layering of the genetic pathways. So that gets a little bit more complicated, but between the black and the chocolate lab, that's what happens for the pigment eumelanin. So how can we work these problems out? And some of you may remember these. These are Punnett squares. What Punnett squares do is allow us to visually represent the sorting of alleles, and that allows us to see all possible offspring based upon the alleles that the parents can give. So, depending on what the parents are, that would then dictate what potential offspring we can make. Now, what I did is I made these draggable alleles, um, and I color coded them. So the mom is going to be this kind of pinky color. Sorry, the dad is going to be yellow. Now, in my first example, uh, we're going to have a chocolate lab mom and then a black lab dad. That's very hard to say. Now, in this case, I'm going to make the dad have a capital and a lowercase. Now, you guys could pause right now and decide as a class what would we call that if it's a one big and one little, meaning that it's different alleles. Now, the mom is a chocolate lab that is recessive. So the only way the mom could have chocolate, the chocolate lab coat, would be as if she is a little b. And again, you guys can pause and decide, okay, what are we going to call that chocolate lab? So if you guys discussed it, you should realize you would say that this black lab is a heterozygous. So the chocolate mom, the chocolate lab mom, would be homozygous recessive. Now, the way the Punnett squares work, you're going to set up one side of the square is is given to just one parent. So the left side is only going to be for dad, and the top is only going to be for the mom. Now, in years past, sometimes they're, oh, you put the mom on what on the whatever. I've not seen books do that. You just want to make sure that one side is given to one parent. The other parent gets the other side. So each row or each column is going to represent one allele that the parent could give. So the black lab dad could give the dominant allele, or that black lab dad could give the recessive allele. So those are the two possibilities for his um, contribution to the offspring. He can either give a dominant black or the recessive chocolate allele. Now, the mom, let's go up and pull hers down. The mom could give the recessive allele, or she could give another recessive allele. Now, a lot of students at that point, they're like, well, that's the same. Yes, in meaning, but there are physically, remember, two chromosomes. So for her, that means she's got one chromosome with the chocolate lab allele, another chromosome with the chocolate lab allele. Now, you could simplify this Punnett square and only do one column. That mathematically simplifies it, but we still would say she has the ability to give one or the other. Now, with the way the Punnett squares work, what you do is you drag across and drag down. Now, I'm going to pull from my top letters just so I don't want to lose what I have on the top and the side. So the dad could give its capital B or capital B, or he can give the lowercase in the bottom. Now there are rules about what goes first, but because we're only seeing little b's on the bottom, it doesn't matter at this point. Capital letters are always shown first. So we have filled in, we have dragged across for the side of the Punnett square. We're going to drag down the top, and so this is going to be one of the offspring, and we're just going to keep going. And if you want to separate, you can move these so they're a little bit more clumped together. That's fine. I like the fact that I color coded mine, which should help you kind of see, oh, that's what mom can do and that's what dad gives. But when we do the Google Doc for homework, you're not going to necessarily see, you're going to have them kind of written together. Okay, that's going to bug me. There we go. So these are all of the potential offspring that we could make. Now that is not a guarantee. So probability means that um, um, it's going to be close to this, just like flipping a coin 10 times. You're probably going to get close to 5 and 5, 5 heads and 5 tails, but that doesn't mean you are going to get 5 heads and 5 tails. But what we do tend to see is in terms of the probabilities, um, the notice we did not get any capital B, so that's 0. The heterozygous, capital B, little b, we see 2 out of 4. Now you can reduce that to 1/2. Now some students love percentages. I'm a fraction person, but if you wanted to write 50%, knock yourself out. That's fine, as long as you can understand the fractions. And then, sorry, this should be little b, little b. So little b, we have 2 out of 4 as well. And again, 50%. You can also reduce it to 1/2 if you want. Again, if we had a litter of puppies, like let's say we had 8 puppies, that does not mean that 4 of them are going to be black labs and 4 of them are going to be chocolate labs. What that means is that every time that mom every time that they created a puppy, there was a 50% chance that it could be a black lab or a chocolate lab. Now, if she has a litter of 10 puppies, it's probably going to be close to that, like 6 and 4 or 7 and 3, but it doesn't have to be 5 and 5. These probabilities refer to the likelihood of getting these combinations. So I made another problem, and then you guys are going to try your hand at some practice. This time, the black lab is the mom is a black lab, heterozygous, and the dad is a black lab as well. So try your hand at this problem, and then you will go over it together in class.